Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, TuneIn Radio, GoodPods. Whatever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro. I am on X as Let's Talk Micro 1 and on LinkedIn as Luis Plaza. I also have an email where you can direct any feedback, any questions, any suggestions, which is letstalkmicro@outlook.com. So please go ahead and subscribe to the podcast, download episodes, and leave a review if the app allows you to do so. As always, any feedback, any suggestions, they are always welcome and appreciated, and thank you for the support. And as I am recording this, I just came back from the Clinical Virology Symposium from the American Society for Microbiology that took place from September 9th through the 12th in West Palm Beach. And it was a great event. I mean, it was my first time attending it. It's definitely smaller than Microbe. I mean, I didn't know what to expect because I hadn't been there before. But overall, it was a great experience. So it was more concentrated on attending the talks and connecting with vendors and connecting with other people in the field. So shout out to Dr. Benjamin Pinsky and other organizers of the event. As always, in this type of events, it is always great when I see podcast guests and I connect with fellow microbiologists. So I got a chance to see some people that I had interactions with before. So thank you, everyone. Those of you that I got to spend some time with you and connect, and it was a great experience. So I'm very thankful for that. And like I said before, I'm planning on doing a short episode about my experience there, and that's just stay tuned for that. But overall, a great experience. And if you haven't listened to the previous episode, please go ahead and do so. It is part one of a two-part series with guests Dr. Ryan Relich from the Indiana University School of Medicine and Dr. Benjamin Pinsky from the Stanford University School of Medicine, two accomplished virologists, very knowledgeable and very passionate. So it was just like listening to an awesome lecture where they go back and forth, and their passion is so great. I always love that when I listen to people that are very passionate about what they do. So it made it such an interesting episode. So on this first part that was published last week, they start talking about viruses, emerging viruses, and they start breaking it down, what type of viruses like DNA, RNA. They talk about diseases, they talk about treatment. So it was a very educational, very informative episode. So please check it out if you haven't already. So today's episode is part two. So the conclusion of that great talk that I had with Ben and Ryan. And in this one, they focus more on diagnosing, on diagnostics, and they talk about testing for these viruses. And they also talk about some of the challenges when you're developing tests, right? Like acquiring samples and things like that. So definitely tune in and check it out. And like I said before, if you haven't checked out the first one, go ahead and do so. But, great information. And at some point in time, we'll definitely have both of them on. Like I said, they know so much and they like sharing information. So definitely stay tuned for future episodes where they come back as guests. And you're going to hear that, like I mentioned on the previous episode, so the Clinical Virology Symposium passed already, but Dr. Benjamin Pinsky said that there's an arbovirus workshop that's upcoming in December. And I have included the link in the show notes. So check it out if it's something that interests you. So let's go ahead and listen to it.
One thing we haven't talked about, and maybe both Ryan and I can tackle this, is how we diagnose these infections. And that's a really important aspect of this. And that's what we do on a day-to-day basis, is make sure that the common viruses we are able to diagnose, but then also, how do we make sure that when something spills over, when there is a human outbreak of these new emerging infections, how are we able to identify those patients? And that's a huge challenge. And a lot of it relies on having very strong public health and epidemiologic surveillance for the emergence of new pathogens and new, foci of folks with new signs and symptoms that are not explained by currently circulating viruses. And it seems like these infections arise in places where perhaps the public health infrastructure is not as strong as it perhaps could be for all sorts of reasons. Investment in low resource settings by high resource countries, the primary reason. But this is a huge challenge. And once these outbreaks are identified, then you have to identify the pathogen. We have those tools currently, and even more so now after the COVID pandemic, we have the ability to sequence pathogens either through metagenomics, where we sequence everything in a clinical sample and identify with bioinformatic techniques new pathogens, or via things like Ryan's really good at, culturing these isolates, which actually has frequently been the way that new pathogens are identified. Even in the modern era, this is how these new viruses are frequently identified. They're thought to be something else sometimes.
Absolutely. And, cultivation-based techniques are, many have written them off as sort of historical relics that still adorn the hallways of some public health lab and reference labs. But, cultivation-based diagnosis, although rare these days, still has its places. And it's especially important for procurement of viral isolates to do further research. All studies that are required to assess the efficacy of antiviral countermeasures, whether they be therapeutics or preventive things like vaccines, you need to challenge either cells in a dish, as well as animals and so on and so forth. And all of that requires virus grown in culture. Even enrichment prior to doing sequencing, if you increase the biological burden of virus within the sample, you have a better chance of your sequencing to turn out nicer sooner. Not to say, though, that doesn't carry some inherent risk with it, because, we had just mentioned that the quasi-species component of RNA viruses. So you're getting a number of viral genotypes that may differ just slightly. There may be this pool of variants, and so you're generating a lot more of those by growing the viruses in culture. So in order to create virus isolates that are fairly closest to what you might see within a person, we only culture them maybe a couple of times. We grow up large volumes of just one or two passages from the primary isolation, so that you don't artifactually introduce adaptive mutations from the monkey cells or whatever that you're using to cultivate the virus. But
Yeah, that's a really important point. And certainly for all the downstream things that you're talking about, you want to make sure that the virus you're looking at is the virus that is the one that's causing problems in the human, not the one that's been adapted to the various cell culture lines that are being used. And certainly we've been asked to sequence all sorts of CoV-2 isolates that have been passaged to make sure that they're still representative of the variant that they put in, rather than picking up various mutations that may impact the interpretation of the experiments that are being done.
Yeah, absolutely. So one of the big biases that has been introduced as a consequence of exactly just that, a lot of people are betting their SARS-CoV-2 data on viruses that have been serially subpassaged. And, they're writing papers about pathogenicity and virulence and other things using, for lack of a better word, attenuated virus isolates. So SARS-CoV-2 has this furin cleavage site that links the S1 and the S2 domains of the spike glycoprotein. Well, after just maybe one passage in Vero cells, you start to lose that, and quite significantly. So after purification of a virus isolate after 3 passages in Vero cells, what you're left with is the vast majority of the progeny particles no longer possess that furin cleavage site. And so if that furin cleavage site contributes to the pathogenicity or virulence of that virus, then what you're using for your experimental model, in my opinion, is no longer relevant.
Yeah, exactly. Yeah, so I mean, obviously we're in agreement, but there's still a huge role that culture, the ability to culture, and specifically the ability to culture organisms that require higher levels of biosafety. This is important in the response to emerging infections because it allows all the things that Ryan is talking about. It allows you to get accurate sequence. It allows the isolates to be used in many, many downstream applications. That's important to our understanding of these infections and also our clinical response. And then the other part of this is like once you have that sequence, you have to make a diagnostic test. And many of these infections, it's a nucleic acid amplification test, usually PCR, but not necessarily. But designing those and implementing them, as we've learned from every single outbreak over the last several decades, is not trivial. And rolling this sort of testing out so that all the patients that need to be evaluated for infection with these agents is also not a trivial task. So that's I guess that's where Ryan and I come in also, is that we have to help with that. And certainly, there's roles that public health is critically important in that, as well as diagnostic manufacturers, because there's no way that clinical labs or public health labs can sustain testing over the course of, if something does become a pandemic, we're not manufacturers. We can't test at that scale without help.
Ben and I, at the beginning of it was, I guess, more like midway through the summer last year. He and I were among the few labs out there that had, number one, the ability to culture monkeypox virus to create reagents that we need to develop diagnostic tests. And so Ben and I worked together. He supplied me with a sample from his laboratory that I took into BSL-3. And I cultured, I grew up a bunch of monkeypox virus, was able to inactivate it, purify the nucleic acid, and then provide that to other laboratories that needed material for assay development, validation, and implementation purposes. So, I was able to procure a bunch of isolates from domestic patients here in Indiana as well. Ben and I have plans for some of those other viruses in the future too. I think he and I want to be, at the front line for our brothers and sisters at arms in the diagnostics field, and be able to at least provide on a smaller scale access to the reagents that are necessary to develop these tests.
Yes, absolutely. And it's important that there are laboratories like Ryan's that can isolate these viruses and make up these materials that can be used for further analytical validation of these tests.
There's not a lot of testing like that, is there? I mean, when it comes to those viruses and
the testing for esoteric viruses is fairly limited. It's relegated to large public health laboratories like Centers for Disease Control and WHO reference laboratories around the world. But for the most part, a lot of the emerging viruses, most clinical labs can't get their hands on the tests. And even if they had the tests available, how would you validate, something like that? A laboratory developed test for Marburg virus, for example, where do you get material other than perhaps, synthetic nucleic acids that you could spike into clinical specimens? That's all well and good, but regulations require that we do our best to secure, actual clinical specimens. And I don't know about you, but I don't have any blood or urine or anything from somebody with Ebola or Marburg in the freezer.
Yeah, and probably, probably all clinical labs or even academic medical centers, don't need to have an Ebola or Marburg test on standby. It's an unlikely to occur. But it's when these things do occur that we need to have a response that is timely, and gets all these different stakeholders together and able to respond and develop these tests and distribute them and implement them in a fashion that allows the best possible care of the patients. And this in the past hasn't happened, but hopefully in the future, what we learned from COVID-2 will be applied to, subsequent outbreaks. And I think there were signs of this for the Mpox story, at least in the United States, and that more labs were able to get tests up and running. The requirements by FDA were less onerous, and I think that the response was relatively quick compared to what occurred with SARS-CoV-2. So certainly progress has been made, and we just got to keep building on that. The one concern is that, as nobody wants to even think about COVID-2 anymore. And as these things fade into the past, there has always been these cycles of interest and then complete lack of interest, which then results in lack of funding, support, and so forth for all of the infrastructure that's needed to monitor for these emerging infections. And so hopefully we won't lose that, because it is inevitable, I think, that these emerging infections will cross over into humans. And, we don't necessarily know which one it's going to be. we have our hypotheses, but we're likely to be wrong. And so we have to be ready with the infrastructure that when one does come in and we're able to identify it, that we can then roll out all the testing so that we can limit the damage to people.
Yes, definitely.
So that was my soapbox for now.
Oh, thank you. It's great. I've been listening here, and I'm very engaged. It's just, this is definitely great. You were mentioning that, I know that Dr. Relich, you mentioned a virus that you have, and this is kind of for me to look it up later and do a little more research, and then whoever in the audience wants to do it as well.
Whitewater Arroyo.
Whitewater Arroyo. Okay.
Yeah, I believe it's named after a state park in New Mexico. Yeah, it's a mammarina virus. So the arenaviruses underwent some nomenclatural revision. Arenaviruses.
Don't get into the taxonomy.
I'm not going to get into taxonomy.
We'll get hate emails.
Yeah, no, I'll stay away from that. But at any rate, yeah, there's an arenavirus called Whitewater Arroyo that's found here in the United States that was associated with 3 cases with 100% case fatality. In each of those cases, Whitewater Arroyo virus was discovered in clinical specimens from the patients, and at least 2 of them died with hemorrhagic fever. So that's some of these viruses are in our backyard. 2 viruses that I study are, one is definitely found in the state of Indiana. It's called Heartland virus, and the other's probably here. It's called bourbon virus. I really like tick-borne viruses quite a bit. And so that's one of the areas that my research lab studies is, sort of the biology and ecology of Heartland and bourbon virus. And I could probably go into the woods, down the road from my house and find infected vectors if I looked hard enough.
And if I remember the Heartland virus story correctly, it was the first patient was initially thought to have ehrlichiosis, and the virus itself was cultured.
Yep.
Because they were looking for Ehrlichia, and it was cultured in the mammalian cell line, and lo and behold, there was viral cytopathic effect.
Yeah. Incidentally, around the same time, a virus emerged in Asia, in parts of Eastern Asia. It's called SFTSV, or severe fever with thrombocytopenia syndrome virus. It's another tick-borne Fenui virus that's very closely related to heartland virus. It appears to be spread by what's called the Asian longhorn tick, or Haemaphysalis longicornis. And incidentally, that same tick is now an invasive species in the United States, but none of the longhorn ticks that we find here on stateside have been shown to be infected with SFTSV. But yeah, those viruses emerged around the same time as one another, and bourbon virus came a little bit later. And that virus was isolated from patients that lived in Bourbon County, Kansas. It's got nothing to do with the viruses being found in bourbon distilleries or anything, and everything to do with their geographic site of detection from Bourbon County, Kansas.
Let's make that clear, because maybe there'll be some sponsorship from one of the bourbon.
Yeah, exactly.
Not virus related.
Not virus related. Nothing to do with the beverages. With regard to naming viruses, people always ask about that. A lot of them have geographic names, or they're named after people. But in 2015, the WHO has tried to put the kibosh on that for a couple of reasons. Chief among which is the fact that naming viruses after places and people doesn't do a whole lot of good for the places and people after whom or after which those viruses are named. People become very suspicious of those areas. Oh my goodness, it's Ryan Relich virus. Don't go anywhere near that person. But so there's stigma that's associated with it, which is why we have names like severe fever with thrombocytopenia syndrome virus and SARS-CoV-2, rather than calling it after the place where those viruses were originally described.
Yeah. Although JC still has the guy's name associated with it. I wonder when that's
sure does
change.
No, I think it's going to be difficult to go back and revise all of those names. I mean, Ebola virus is named after the Ebola River in northern DRC. Marburg virus is named after Marburg, Germany. A lot of these high consequence virus names that everybody's read about, either in a fiction or a nonfiction or a mostly nonfiction book, they come with names from wherever they were originally described.
As we're talking about this, so we're also talk about outbreaks, and we see a lot of information out there, and it's always good, right, to talk about it in a responsible manner. And so as far as what viruses are we seeing in outbreaks, and typically when we think about them in the public, we think about that, right? Where are they taking place? What's causing it? Is it under control? Is it on the increase? So what viruses are we seeing on outbreaks lately?
I'm going to answer on behalf of both me and Ben when I say dengue virus. That's one right now that seems to be absolutely everywhere. Lots and lots and lots of headlines about unprecedented outbreaks of dengue fever throughout Asia and Latin America.
Yeah, absolutely. So dengue is a huge, huge problem, and there seems to be a post-pandemic increase in the number of cases. So dengue is a really important virus, and has been important for decades now. A virus that there are no therapies. There is a vaccine that doesn't work particularly well, and requires that someone be infected at least once before they get the vaccine. So there's a lot of work to do there, but a lot of interest now. So hopefully there will be some progress there. And actually, I'm on a committee that is putting together a symposium on emerging arboviruses, so viruses transmitted by arthropods like mosquitoes and ticks. And so that's going to be this December through the National Academies of Sciences and National Academy of Science and Medicine. Science, Engineering, and Medicine, if I get that correct. So that should be really interesting, because I think it's not only dengue, but any of these viruses that are transmitted by arthropods, the problem is that with our climate change, that the vectors are spreading. They're essentially everywhere. And this allows us to come into contact with these vectors. So it's a really interesting and concerning area.
Most definitely.
The other viruses, there's a lot of concern over the past several years about avian influenza. I think Ryan mentioned those earlier, but there's a particular clade 2344B that has spread throughout the world and has caused the deaths of tens of millions of birds, both wild birds and farm poultry. So fortunately, there has not been very many cases in humans, but this is certainly something that folks are watching closely.
Yeah, and we're even seeing viruses crop up in, quote, new places. For example, Crimean Congo hemorrhagic fever virus, CCHFV. Yeah, that's a tick-borne virus. Hyaloma ticks or camel ticks are associated with its transmission. And in the past couple of decades, we've seen new foci of infection emerge in Spain. Eastern Europe has a long history, I think, with CCHFV, but hasn't really been until the 21st century that we saw the emergence of CCHFV in Western Europe, which is scary.
Yeah, there's a lot. The vectors are spreading, and the viruses associated with them are also spreading. And so this is something that is going to continue to occur as the ecology of these organisms change.
Yep. And some of them, we don't have any good control strategies at the moment. We can do our best to avoid the vectors of them or change our lifestyle a little bit, but not everybody follows the rules. Some people don't believe in the science. There's nothing to not believe in because it's a fact. You can believe or disbelieve all you want, but that doesn't change the facts. And so you really need everybody to participate. And so, as a function of that, we see these hot spots crop up every once in a while. It's people doing things perhaps they shouldn't be doing, like with Nipah virus in Bangladesh and India. Harvesting date palm sap that's not boiled or heated in some way to destroy the virus. Those people will go door to door selling their date palm sap that's potentially contaminated with teropid bat excretions containing Nipah virus, and so you then create an outbreak. However, safe food handling practices, cooking the date palm sap prior to consumption would do a lot for public health, but that's not done. Maybe it ruins the flavor. I don't know. I've never tasted date palm sap that I know of, and maybe raw is better than cooked. But it's sort of the same logic with pasteurization, right? I mean, with pasteurization, you see many fewer cases of bovine tuberculosis and brucellosis and other things that can be transmitted in the milk of cloven-hoofed animals.
Yeah, Nipah is a really good example, and that's one that you and I have talked about as being one that we're concerned may spill over into humans even more so than it already does. It's a scary infection, results in many cases fatal encephalitis. So that's one where there's concern that it could become more transmissible and therefore cause greater outbreaks, and that could be devastating.
You mentioned that there's going to be a symposium, Dr. Pinsky's out for the arboviruses.
Yeah, it's I can send you afterwards the dates, and maybe you can put it linked with this. It's being organized right now. I believe it's the second week of December of this year. But it's currently being organized. We're inviting speakers, and it should be really interesting.
Okay, so yeah, definitely, and as the listeners, if you're, I will, once I get the information, I will put it on the show notes, or As it gets updated, I will put it on social media as well. And if that's something that interests you, that way you can look it over.
Yeah, great. That's awesome.
Yeah, and you saw there's also you said in August there's.
Oh yeah, in September there's the Clinical Virology Symposium. That takes place in West Palm Beach, Florida. Registration is still open. This is the main clinical virology meeting in the United States. Typically have about 800 to 1,000 people interested in virology, and it's all aspects of clinical virology. One of the sessions, like I mentioned, is what Ryan and I do is clinical virology Jeopardy, which is always a lot of fun. There's a session on case reports. There's a whole practical session about diagnostics, also epidemiology and therapeutics. So everything is really covered. And I can say it's run by American Society for Microbiology, and I'll also send you the link to that, and so people can check that out and potentially attend in person. It's a really good lineup this year. But of course, I am a bit biased because I'm the chair of the organizing committee. But it should be really good.
Okay, yeah, definitely. Please do, and for the listeners, I'll also put that on the show notes. That way you can look it over, and then if it interests you, if you want to sign up or get more information, you can go ahead and do so. Um, I could listen to both of you talk for hours, and this has been great. Is there anything else that you want to add?
There's probably a lot, but for the sake of your listeners not turning us off, I'll just say, just definitely check out the links that Ben is going to provide if you're interested in either of those conferences. Certainly go. The science is about communication, science is about experimentation, science is about learning. And so one of the best ways to do that, I think, is by networking with other scientists and other people who are interested, and even reaching out to medical laboratory personnel and other folks, other stakeholders. We're all on the same team here, and so we need to network. We need to get to know each other. We need to work with each other. And one thing that I would love to see is more interactions between public health laboratories, diagnostic, clinical laboratories, and even academic laboratories working together to build capacity to deal with future, viral outbreaks, and not even just viruses, but bacteria, fungi, parasites, prions, so on and so forth. And so, I mean, that's totally necessary if we want to stay ahead of these things. And so, yeah, with that, I'll get off my soapbox and leave it up to Dr. Pinsky.
I've already been on my soapbox, so I agree with all that you just said, and virology is a really interesting field. There's so much that there's so much to learn, and there's so much impact we can have on humanity and the world itself. So yeah, I really enjoyed talking to both of you. And yeah, check out those links. And if you want to attend any of those, I encourage you. And certainly Ryan and I are available and easy to find if there are specific questions that folks have.
Yeah, absolutely. I don't mind answering emails and other things. Ben and I, the last thing that I'll say, we're in the process of planning an expedition to Zimbabwe to do some emerging virus surveillance for another arena virus called Lujo virus. And so maybe if, whenever we're able to get that done, we'll send you some pictures you can post on, on your social media, Louise.
Oh yeah, that would be awesome. Probably sometime next spring. I think that's the plan. But yeah, we're looking to do our own little surveillance project on that particular virus. But yeah, that should be really fun.
Yeah, I will love to hear about it. So if you're okay with come doing an episode about it where you can talk about
Yeah,
yeah,
we can talk about our visit and what we found. That would be a lot of fun.
yeah, that will be great. So definitely, like you have said, communication and networking, and it's such a great era where we can be so connected, and with social media, and so it's just that's how I met, I have met people, and you can see publications and reach out to people, and pretty much in everyone in this field, most people have met, they're very friendly, very reachable, like both of you are, and they just want to talk about what you do, and you want to, like I say, get in your soapbox and talk and share your information, and I love that. I love the passion and the work that you're doing. It's and I said this before in other episodes, but it is great when you see people, right, that they use their knowledge, their education, and their work, and just for make this feel better. So I appreciate what both of you are doing, and thank you, for taking the time and coming into Let's Talk Micro.
Absolutely. And right back at you, my friend. I mean, Ben and I are big fans of yours as well. You're doing something that Ben and I haven't been able to do, which is, have this successful podcast of bringing on all these thought leaders and experts and so on and so forth. You're getting the information out there, and you yourself are also a very talented microbiologist. So it's, that's the pleasures all on this side of the table, I think. So thank you very much.
Yeah, thank you.
I appreciate it. Thank you so much. And that, my dear audience, was the end of this episode. I hope you enjoyed the conclusion of that great talk about viruses with Dr. Relich and Dr. Pinsky. As always, I enjoy sharing this information with you. So please continue bringing that passion to what you do. It's so important. You do such great work. And stay tuned. Great episodes coming your way. As always, thank you for the support. So stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.